Histological sections. Histological sections are thin layers of tissue prepared and observed under the microscope for the study of the structure, composition, and characteristics of biological tissues. These sections are crucial to histology, which is the study of tissues, the diagnosis of diseases, the understanding of biological functions, and the research of various conditions. Here is the step-by-step preparation and examination process of histological sections. 1. Fixation. Purpose: fixation maintains the tissue structure and prevents degeneration. It arrests enzymatic reactions, which may break the tissue, and keeps the tissue in a lifelike state. Common fixatives, formalin and formaldehyde are the most common fixatives. Alcohol and glutaraldehyde are the other fixatives. Process: the tissue is left in the fixative for hours or even days, depending on its size and type. 2. Processing. After fixation, the tissue is processed to remove water and replace it with a medium, usually paraffin wax, that will support the tissue for slicing. This process includes dehydration. The tissue is subjected to a series of alcoholic baths with progressively higher concentrations to remove water. Clearing: alcohol is replaced with a clearing agent like xylene that makes the tissue transparent and ready for embedding. Embedding: this refers to the infiltration of paraffin wax into the tissue, which provides support to the tissue and facilitates ease in slicing into very thin sections. 3. Sectioning. Microtome: thin sections of the embedded tissue are sliced using an instrument called a microtome. It is usually 4 to 10 micrometers in thickness. Thickness: the thin slices allow light to pass through the microscope for examination. 4. Mounting. The thin sections are placed on glass slides. In some instances, the paraffin may be melted out, if necessary, and the sections are allowed to stick to the slides, often with the use of an adhesive. 5. Staining. Goal: tissues are typically nearly invisible under a microscope. Staining offers contrast in color to specific parts of the tissue so they can become more easily visible. Common stains: the most common stains used are the following. Hematoxylin and eosin, H&E nuclei are stained blue or purple with hematoxylin, and eosin imparts pink color to the cytoplasm and extracellular matrix. Special stains: these stains demonstrate specific tissue components. Examples are the following. Masson's trichrome for staining collagen. Periodic acid Schiff, PAS, for staining carbohydrates. Silver stains for staining nervous tissue. 6. Microscopy. Light microscopy: after the tissue sections are stained, they are viewed through a light microscope. A microscope increases the magnification of the structure, and, therefore, a researcher or a histopathologist may note characteristics that are not visible to the naked eye, detect abnormalities, or appreciate its architecture. Electron microscopy: if needed, if the researcher intends to get further details about the cellular structure, then the section is prepared for electron microscopy, which yields much clearer images than the light microscope. 7. Interpretation. The stained sections are examined by histologists, pathologists, or researchers to interpret the tissue structure and identify abnormalities such as inflammation, cancer, fibrosis, or any other pathological conditions. Examples of analysis: identification of the shape and size of cells and how they are arranged. Identification of various types of tissues, epithelial, connective, muscular, and nervous. CSS pathological variations or alterations, i.e., distinguishing cancerous from non-cancerous cells. Applications of histological sections. 1. Clinical diagnostics: histology is applied in pathology while diagnosing diseases and diseases' onset and cause. This goes beyond merely ascertaining whether it is a case of cancer, infection, or autoimmune disease, among others. 2. Research: histology allows scientists to study tissue morphology and physiology, hence, it makes it easier to understand normal biological processes and disease mechanisms. 3. Education: medical and biology students use histological slides to learn how human and animal tissue operates. In one word, histological sections have pictures of microanatomy in detail and become the best tool to understand tissues under normal conditions as well as when in the diseased state, contributing the greatest possible part to diagnostics and research.